Hello everybody, today we are going to talk about pathophysiology of the nervous system. We'll discuss involvement of neurons and glial cells in variety of disorders in the nervous system. I want us to acknowledge and differentiate astrocytes, oligodendrogliocytes, ependymal cells, and microglial cells as important cells within the brain. And a short introduction to neurotransmitters, because we will discuss mechanisms related with them and with their deficiency, or when we have too much of specific neurotransmission. So first we address the question, what is a neurotransmitter? So classically, a neurotransmitter is a small molecule, usually derived from amino acid, and it is localized in the presynaptic terminal, and it is in its specific vesicles. And when action potential arrives, voltage-gated calcium channels are opened, which results in activation of a molecular machinery, which eventually results in release of neurotransmitter from the vesicle into the synaptic cleft. And finally, it should arrive at postsynaptic membrane, where it should have its receptor, and application of agonists or antagonists should mimic the effect of neurotransmitter. So if all these three conditions are met, we may consider given molecule a neurotransmitter. But in addition to typical neurotransmitters, we have also involvement of so-called co-transporters or co-transmitters, which are usually peptides, chemically, structurally, and they also are found in vesicles, not in all neurons we have them, but they are usually localized a bit far away from the postsynaptic, presynaptic membrane. Here they are shown in blue. And for providing an example and at the same time to show the role, let's consider one of them as substance P in these blue ones, and glutamate in green ones, so that let's consider that this is a synaptic contact in the dorsal horns, where we have the first contact of pain transmission. So given this context, if we have low frequency stimulation, we have majorly and preferentially release of glutamate only, the small molecule transmitter, and the substance P is still here, not released. But if we have higher frequency stimulation, when we have more diffuse increase in calcium concentration within the presynaptic terminal, the result is release of both types of neurotransmitters. And also we should appreciate the fact that substance P is released not only into the cleft, but also, as it's shown here, it's released in, like, let's call it paracrine manner, so that this would modulate efficacy of other neighboring synapses. And to add to this, we should also appreciate the fact that most neurons usually we consider as mono transmitters. I mean, we may say that this neuron is a glutamatergic neuron, so it would mean that it releases glutamate, and so it means that most likely it is an excitatory neuron, as opposed to some GABAergic neuron, which would be correspondingly inhibitory neuron. But in reality, it's shown that some neurons have two or even sometimes three different small molecule neurotransmitters, which add some complexity in understanding these neurons and their networks, which we maybe should compare to our languages. Like, all of us usually have one language that we know better than the others, and it is our mother language, like Armenian for me. but most of us also have ability to talk and understand some other languages as well. So we may consider that this similarity exists in neurons as well. They may have some neurotransmitter which is classical one and its major type, but others also add some additional value to this neuron and increases the complexity of the network. To continue the discussion of transmitters, we should also show some examples when different transmitters are involved in different problems and diseases. Like the classical neurotransmitter acetylcholine is found absolutely everywhere, so in the brain, in the peripheral nerves, and it was the first studied classical neurotransmitter, and depending on where it is and what kind of receptor we have, it could have both excitatory or inhibitory effect. We know that it is its activity is decreased in Alzheimer's disease. We probably should also remember example of myasthenia gravis, in which we have antibodies against acetylcholine receptors at the postsynaptic terminal neuromuscular junction, so it is in the periphery. The problem is in the periphery, and we have again the problem related with cholinergic neurotransmission. Another example could be botulotoxin, which also affects cholinergic system. The other examples are also given here. I don't want to lose time in this short video to discuss all these things. But common examples of dopamine, I think we should add also, of course, the most common it's problem, which is Parkinsonism or Parkinson's disease, when dopaminergic neurons in substantia nigra are degenerating faster than others, which results in decrease in corresponding regulatory effect of dopamine system. And interestingly, dopamine, despite the fact that it's written or mentioned here that is usually excitatory, we should consider that it has no ionotropic receptors. Instead, it has different types of metabotropic receptors, both inhibitory and excitatory, which allows dopaminergic neurons to be involved in very interesting regulatory, smooth tuning activity of the basal ganglia and so-called extrapyramidal system. Nevertheless, I want to remind you also the idea of non-classical transmitters. In this sense, they are non-classical since they act not from pre to post but in opposite direction. For instance, in the glutamatergic neurons, we have also such feedback when glutamate is released, and if it activates through NMDA receptors, about which we'll talk a bit later, entrance of calcium, so that nitric oxide synthase is activated, and it results in production of nitric oxide, which diffuses back to the presynaptic membrane and allows more neurotransmitter to be released, so that it allows more wiring together of these two neurons by increasing the weight of this synapse. We have also opposite working transmitter, an example of which is tetrahydrocannabinol, and it has similarity with the nitric oxide idea, but in opposite manner, so that here postsynaptic neuron, by releasing tetrahydrocannabinol, would decrease release of transmitter, thereby preventing overactivation of the postsynaptic neuron. And this activity is especially important, of special importance for pain neurotransmission, for instance. That's why cannabis sometimes is medically used for treatment of pain, which is not controlled otherwise by different narcotic analgesics. Instead, sometimes, especially in cancer pain, this becomes an important option, as well as such back inhibition. If we can call it like that, is important in treatment of at least some types of epilepsy, when overexcitation of neurons and firing there together results in significant damage to the brain, so that sometimes cannabis consumption allows treating the epilepsy and preventing seizures. Since the most important neurotransmitter for us in the brain would be glutamate, let us discuss the structure of receptors having this example. So in general, neurotransmitters may have either ionotropic or metabotropic receptors. For glutamate, metabotropic receptors are called mGlu-Rs, from 1 to 5. There are different metabotropic receptors with different localization, but anyway, we appreciate that it works here as a in a way that hormones usually work, through corresponding receptors. While ionotropic receptors are coupled with corresponding channels, and for glutamate we have AMPA and kainate receptors, which are permeable for sodium ions, and we have elegant NMDA receptor, which is permeable both for sodium and calcium ions. But importantly, under normal conditions, NMDA receptors are blocked by magnesium ions, which make this channel impermeable. But if we have increased stimulation of AMPA receptors and increased entrance of sodium into the cytoplasm, it results in depolarization, which kicks out this magnesium block and which results in permeabilization of NMDA receptors, which is followed by influx of sodium and calcium ions. And this is probably the most important event for understanding the idea of having more than one ionotropic receptor. Slow activation, slow signaling, only AMPA and only sodium, significantly calcium adds in. So what it does work for? It has its important implications in physiological and pathophysiological stimulation situations. Normally in physiological aspect, though, it is involved and serves as the basic mechanism for one of the most important mechanism of synaptic plasticity. And here how it works, let me show you. So normally baseline transmission results in activation, again, as we see here, of yellow ones are AMPA receptors, so sodium flux without calcium, which is blocked because magnesium ions are here. But intense stimulation results in removal of this block, so we have induction of long-term potentiation, and we have sodium and calcium ions flashing in. The consequence of this is that calcium ions result in several changes, which include increasing efficacy of AMPA receptors, as well as increasing their numbers, because calcium ions, through several steps, overall increase the number of AMPA containing vesicles moving to the cell surface and putting these receptors at the cell surface. So here we see that the number of AMPA receptors is now increased, which will make this synapse stronger or much efficient. So in any way, the idea is that as one of the best neuroscientists that I know, Leonard White, and who is also the author of the medical neuroscience textbook, says neurons that wire together, they are also that fire together also wire together. Let me repeat again. Neurons that fire together, like here these two neurons, they wire together. This synapse is now stronger. Moreover, this could be continued into increasing numbers of dendritic spines, so that we would have more contacts in these two neurons. So this is a mechanism of memory, and once we have activation of several neurons simultaneously, it means that they will code some memory. But unfortunately, the same mechanism becomes activated in mechanisms of central sensitization, in pain mechanisms, or excitotoxicity, which we'll discuss later, is again hijacking this mechanism that we saw here. Now let's turn our attention to the etiology and pathogenesis of damage in the nervous system. The causes we may classify into exogenous and endogenous, and exogenous ones, some of them are specifically neurotropic, and that's why they may require our attention. So by nature, exogenous factors could be physical, chemical, biological, with corresponding examples provided here. But also, let's appreciate psychological factors play important role, because the nervous system is the one that is related with our secondary signaling systems, with our speech, psychological events. All this has its own impact on the nervous system. Out of the mentioned examples, we should also consider that, like bacteria, if I turn my attention to biological factors, are causing damage everywhere, but the other examples are more specific, like prions. They affect, interestingly, only nervous system, at least to our current knowledge. Or we have specific viruses that have some neurotropism in them. It is rabies, poliomyelitis, herpes virus, etc. Out of chemical factors, especially vitamin B1 is known to cause Wernicke-Korsakoff encephalopathy, or syndrome better, where we find different chemicals. More specifically, urare is neurotropic. Out of physical factors, mechanical injury is there. We should appreciate role of a skull and spinal cord here, and it has a dual role. Yeah, it is protecting, of course, our tiny and vulnerable nervous tissue, but on the other hand, sometimes the damage may be caused by the cranium itself. And ionizing radiation, we usually say that the most vulnerable tissues to radiation are those which are rapidly proliferating, like hematopoietic tissue, and it is still true. But on the other hand, if ionizing radiation is severe enough to cause damage to the nerves, so if we have nervous involvement in the radiation disease, it would be very severe and usually lethal if such significant radiation exists. And hypoxia, the most common mechanism and pathological process that we have, is emphasized here again, because again of the extremely high vulnerability of the nervous system for hypoxia, because of the very high metabolic demand of the nervous systems. And that's why some scientists suggested this term pathoclisis, or selective vulnerability, and we may understand the idea of pathoclisis in two ways. First, nervous system itself is selectively vulnerable, but even within the nervous system we have different sets of neurons which are selectively vulnerable towards different pathogenic factors. For example, we know that Purkinje cells are selectively vulnerable for ethanol, where we may appreciate that In hepatic encephalopathy, one of the most interesting symptoms that patient developed was so-called asterixis, and that kind of tremor was associated with increased manganese concentration in the brain, which is actually found to be much more affecting globus pallidus in the basal ganglia, which results in such effects. So this is very interesting story why these neurons are much vulnerable than the others. Another example of this related with the previous slide with hypoxia is the idea that large hippocampal cells and again Purkinje cells are vulnerable to general hypoxia. Well, again, as an example for different neurotoxins which are neurotropic. Let me just skip the intrinsic factors because most of them we know from previous our studies and discussions. Some of them we discussed in more details, like in hepatic encephalopathy for hyperammonemia, etc. Out of metabolic disorders we discussed within the diabetes mellitus topic. Just to emphasize here that neurons almost exclusively depend on, as we know, glucose as their main metabolic source, so that hypoglycemia would be of special importance for the brain. And since we discussed factors that may cause damage, let us go through our defense systems. We defend our nervous system by help of our skull, our vertebrae, which protect from brutal physical force. Next, we can consider and appreciate anti-systems. To understand them, I asked our colleague to explain what is actually Hegel saying here. Yes, and the point is that if I have one system, I would have another system to counteract with, to balance it. And the idea here is that if I have nociceptive system for pain, I have anti-nociceptive system which would smoothen or modulate its effect. If I have so-called epileptic system, which is of course pathological, and neurons which would activate one another and eventually resulting in their death, we also should have corresponding anti-epileptic system, which would be probably a set of neurons that would have inhibitory effect and that would prevent over-activation of several groups of neurons. And overall, the idea of excitatory and inhibitory neurons probably also is fitting within this idea of anti-system. So if the system would be a system of excitatory neurons, the inhibitory neurons would be there. And the idea why we discuss this in this way, of course, it's some artificial to separate system versus anti-system, and maybe Hegel would appreciate it more than others, but the point here is that sometimes the damage is associated with anti-system. An example is that if we have degeneration or damage to Rancho cells, which are inhibitory neurons in the spinal cord, the result is that we have liberation, I would say, from this inhibitory control of alpha motor neurons, and the result is tetany. And that was the mode how tetanotoxin acted. It entered actually and caused impaired release of inhibitory transmitters from the branchial cells in the spinal cord. Separately, we'll discuss blood-brain barrier, meninges, membranes, and let's also appreciate that brain has separate status in sense of immunity, and it has long been considered as immune privileged organ in sense that most of T or B cells normally are not allowed to enter the brain tissue to scan there for some antigens, etc., because it is strictly prohibited there. But in pathological conditions, of course, we find there different types of lymphocytes, and unfortunately it was wrong idea to allow them to go into the nervous system. Astrocytes together, they are forming actually their own immune